Baik Bapak Ibu, mengawali acara pertama yaitu sambutan Kepala Organisasi Riset Pertanian dan Pangan BRIN kepada Ibu Puji Lestari SPMSI PhD, kami silakan.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. The Excellency, the head of Center for Animal Husbandry, Dr. Santoso, distinguished speaker, Dr. Muhammad Asanim, the visiting researcher in the Center for Animal Husbandry, respected researchers, academics, practitioners, stakeholders, and all participants of the 10th Risnov Ternak webinar. First, let us be grateful to Allah, God Almighty, for allowing us to gather virtually in good health through this theme, metagenomics and immunology. The future of livestock health management is highly relevant and strategic in addressing the challenges of modern livestock system. Ladies and gentlemen, the livestock sector faces rising demand for safe, high quality, and environmentally friendly animal products, while also confronting climate change, resource pressure, and animal health issues. This requires integrative and multidisciplinary research, advances in nutrition, molecular biotechnology, microbiome studies, and immunology now enable more precise and effective intervention. Functional foods, probiotics, and plant bioactives, and molecular tools such as RNA sequence and microbiome profiling offer new ways to enhance natural resilience, reduce antibiotic use, and support sustainable livestock system. So through this webinar, we hope to further explore various research innovations carried out by BRIN researchers and collaborators. I'm confident that the presentation today will enrich our understanding of how the integration of nutrition, feed technology, molecular approaches, and immunological strategies can serve as a foundation for advancing Indonesia's livestock sector. So Research Organization for Agriculture and Food is pleased because this activity are not merely forums for sharing information, but also part of our effort to strengthen a collaborative research ecosystem. We aim to explore innovation that strengthen productivity, animal health, and sustainability while contributing to Indonesia's commitment to low carbon food system. I encourage all participants to actively engage in this discussion and collaboration as future livestock development demands collective effort across disciplines. Finally, I extend my sincere appreciation to our speaker, organizer, and participant. By saying Bismillahirrahmanirrahim, I hereby officially open the Risnov Livestock webinar or Risnov Ternak webinar seri 10. And thank you for your participation and enthusiasm. May this webinar strengthen collaboration and bring real benefit to the advancement of the livestock sector. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Acara berikutnya yaitu pengantar Kepala Pusat Riset Peternakan Organisasi Riset Pertanian dan Pangan BRIN kepada Bapak Dr. Hewan Santoso MSc, kami silakan.
Baik, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera bagi kita semua. Om Swastiastu, Namo Buddhaya, salam kebajikan. Thank you for Miss Yeti Lestiani SE Com. And sorry, ladies and gentlemen, please allow me to extend my sincere apologies on this occasion. I will be delivering my remarks in bahasa. Okay, yang terhormat Ibu Puji Lestari, SPI, MSI, PhD, selaku Kepala Organisasi Riset Pertanian Tangan, and then narasumber Dr. Muhammad Ahsan Nahem, visiting researcher Pusat Riset Peternakan BRIN. We are deeply honored to welcome you to this webinar. Thank you very much for taking the time to share your knowledge. Your presence truly enrich today's discussion and bring valuable insight for the advancement of a sustainable livestock system in our country. And then terima kasih, thank you Ibu Ezi Masdiani Putri, rekan-rekan peneliti, akademisi, praktisi, pelaku usaha, dan seluruh peserta webinar yang saya hormati dan saya banggakan. Bapak Ibu peserta webinar yang saya hormati, pertama-tama marilah kita panjatkan puji dan syukur kita kehadirat Allah Subhanahu wa Ta'ala, karena pada hari ini kita diberi kesempatan untuk kembali berkumpul dalam ruang virtual untuk melaksanakan webinar Risnov Ternak yang ke-10 dengan tema yang mengangkat Metagenomics and Immunology, the Future of Livestock Health Management. Ini merupakan topik yang sangat strategis dalam memperkuat fondasi kesehatan ternak modern. Bapak Ibu yang saya hormati, perkembangan ilmu pengetahuan saat ini mendorong sektor peternakan untuk bergerak lebih cepat dalam mengadopsi pendekatan ilmiah yang lebih integratif. Tekanan terhadap sumber daya alam, perubahan iklim, tingginya kebutuhan pangan hewani, serta tuntutan konsumen terhadap produk yang aman, berkualitas, dan ramah lingkungan menjadikan inovasi berbasis riset semakin penting untuk dihadirkan. Bapak Ibu, kita sedang memasuki fase baru di mana dalam pengelolaan kesehatan ternak, kemudian pendekatan tradisional yang selama ini menjadi tumpuan, baik dalam pemberian pakan, kemudian pengendalian penyakit maupun manajemen pemeliharaan, ini tidak lagi cukup untuk menjawab tantangan yang semakin kompleks. Nah, disinilah teknologi metagenomik dan imunologi memainkan peran yang penting. Melalui analisis metagenomik, kita dapat memahami komposisi mikrobioma dan hubungannya dengan kesehatan serta performa ternak secara lebih mendalam. Sementara itu, pendekatan imunologi modern memungkinkan kita mempelajari bagaimana sistem pertahanan tubuh ternak bereaksi terhadap berbagai faktor stres, kemudian penyakit maupun intervensi nutrisi dan pakan fungsional. Nah, integrasi kedua pendekatan ini memberikan peluang bagi kita untuk merancang strategi kesehatan ternak yang lebih presisi, baik melalui pengembangan pangan fungsional, probiotik, dan prebiotik. Nah, kemudian pemanfaatan metabolit sekunder tanaman hingga inovasi seperti microbiome modulation dan immune system optimization. Nah, lebih dari itu, Bapak Ibu, pendekatan ilmiah yang komprehensif ini juga mendukung upaya global dalam mitigasi resistensi antimikroba atau AMR, serta perkembangan sistem peternakan rendah karbon dengan kesehatan ternak yang lebih baik, efisiensi pakan meningkat, emisi menurun, dan rantai pasok pangan hewani menjadi lebih berkelanjutan. Nah, melalui webinar ini saya berharap kita dapat menggali pemikiran, kemudian pengalaman dan rekomendasi ilmiah dari narasumber kita, sehingga kolaborasi dan inovasi dapat terus berkembang. Okay, baik, I think that in decision carries an important theme, metagenomic and immunology, reflecting the future direction of livestock health management. In Indonesia, the livestock sector is experiencing rapid transformation driven by increasing demand for safe, nutritious, and sustainable product animal protein. At the same time, we are challenged by environmental pressures, antimicrobial resistance, and the need for more precise and science-based approach to improve productivity. And thank you for Mr. Muhammad Ahsan Nahem. And Bapak Ibu yang saya hormati, sebagai penutup, saya mengajak seluruh peserta untuk memanfaatkan forum ini sebaik-baiknya berdiskusi, kemudian bertanya dan menjadikan pengetahuan yang diperoleh sebagai pijakan untuk memperkuat riset peternakan Indonesia. Akhir kata, saya menyampaikan terima kasih kepada seluruh pihak yang telah mendukung terselenggaranya kegiatan ini. Ini khususnya Dr. Muhammad Ainsar Harahap. Terima kasih Bapak telah mengkoordinir pelaksanaan webinar hingga akhir tahun ini. Kemudian dari Humas, terima kasih dukungannya, para narasumber, moderator, serta seluruh peserta. Terima kasih. Semoga webinar ini dapat berjalan dengan baik dan kita dapat memperoleh ilmu yang bermanfaat. Saya akhiri. Billahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih Bapak atas pengantarnya. Baik Bapak Ibu, berikutnya kita akan melakukan sesi foto bersama. Kepada Bapak Ibu yang berada di ruang Zoom, silakan untuk membuka kamera dan memberikan senyum terbaiknya. Silakan Bapak Ibu untuk membuka kameranya. Baik, saya akan mulai sesi foto bersama sesuai dengan aba-aba saya dan panitia akan melakukan tangkap layar. Baik, saya mulai 1, 2, 3 untuk layar pertama. 1, 2, 3 untuk layar kedua. 1, 2, 3 untuk layar ketiga. Baik, terima kasih Bapak Ibu. Acara selanjutnya yaitu kita akan memasuki acara inti, pemaparan materi dan diskusi yang akan dipandu secara langsung oleh moderator kita pada hari ini yaitu Ibu Dr. Ezi Masdia Putri SPT, Penelaah Teknik Kebijakan Pusat Riset Peternakan ORPP BRIN. Baik, langsung saja kepada Ibu Ezi Masdia Putri, kami silakan.
Baik, terima kasih Bu Yeti atas waktunya. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi dan salam sejahtera untuk kita semua. Yang terhormat Ibu Kepala Organisasi Riset Pertanian dan Pangan, Ibu Puji Lestari SP MSI PhD. Yang terhormat Bapak Kepala Pusat Riset Peternakan, Dr. Hewan Santoso MSI. Yang terhormat Bapak Ibu para peneliti, akademisi, praktisi, serta seluruh hadirin yang sudah hadir di ruang Zoom hari ini. Perkenalkan, saya Ezi Masdia Putri yang akan bertugas sebagai moderator pada hari ini. Hari ini kita sangat beruntung karena dapat menghadirkan narasumber tamu, visiting researcher from Pakistan, Dr. Muhammad Ahsan Naim. It is an honor to have you here with us today. We truly appreciate your presence and look forward to learning from you. Bapak Ibu, Dr. Ahsan akan berbagi pandangan mengenai bagaimana teknologi metagenomik dan imunologi dapat menjadi strategi masa depan dalam manajemen kesehatan ternak. During today's webinar, the event will be followed as a presentation by the speaker for 30 minutes, question and answer session for 45 minutes, and closing. Sebelum memulai acara, izinkan saya menyampaikan beberapa aturan teknis. Pertama, seluruh peserta diharapkan menonaktifkan mikrofon selama pemaparan berlangsung. Dan jika ingin bertanya, silahkan menggunakan fitur raise hand atau tuliskan pertanyaan di kolom chat. Baik, saya akan membacakan CV Dr. Ahsan secara singkat. Dr. Muhammad Ahsan Naim, visiting researcher asal Pakistan, telah menyelesaikan PhD studinya di Huazhong Agricultural University, Wuhan, China, di tahun 2016 hingga 2020 di Preventive Veterinary Medicine. Beliau saat ini dosen di University of Veterinary and Animal Sciences, Lahore, Pakistan, dan beliau memiliki pengalaman sangat luas dalam riset biologi molekuler, imunologi, mikroskopi, bioinformatika, dan penelitian mikobakteri. Tanpa berpanjang kata, mari kita persilahkan untuk narasumber memulai presentasi. Dr. Ahsan, the time and screen are yours, please.
Assalamualaikum and thank you everyone. I'm highly thankful to the government of Republic of Indonesia, first of all, and secondly for all the stakeholders from the BRIN community like Ms. Puji Lestari, Dr. Hewan Santoso, and Miss Yati Lastriani, and also my host from the BRIN, Dr. Easy Masdia. So today, almost I'm here for the 2 months, and it's now the completion time for my visiting researcher program. And I have to present some of the important issues which are going through the livestock sector and how we can advance them, especially from the perspective of Indonesia, to advance their livestock sector, and how we can incorporate all those things together under the same umbrella and same platform. So just for my brief introduction for the audience so that they can easily get to know me, that currently I'm working as a senior lecturer in the University of Veterinary and Animal Sciences, and also as an associate director of student affairs. So regarding my few research expertise, as during my PhD, I explored the genetic role of tuberculosis, and previously my background in masters in veterinary pharmacology. So I'm also a registered veterinary medical practitioner from Pakistan Veterinary Medical Council, and also the registered veterinary faculty. And I also got FHEA, which is actually a diploma to teach at the higher education system in the UK. So some of my research awards is 2 times from the Bill and Melinda Gates Foundation in 2024 and 2022, and also once from the OIC, which is the Organization of Islamic Countries, in 2023. And right now, almost I have more than 23 research papers and more than 8 review papers, and several are ongoing. And 3 are the clinical case reports, 1 full book, and several are the chapters. So here is brief introduction of my university. It actually consists of 4 campuses in different cities of Pakistan, and it is a W category university, which means that it is the high or top ranked university in the veterinary sector. So just jumping on to what we have done up till now. So I will explore here, or you can say this, summarize few of my things previously we have done in Pakistan, and also then taking back to here in the Indonesia. So in Pakistan, we started a project in 2022 with the help of funding by the University of Veterinary and Animal Sciences regarding to explore the herbal medication development from various locally existing plants. So what we do that we took actually so many, but out of them, these 6 were the top ranked, and we conducted the GCMS to check their more active ingredients, and then we check their ability to in the Salmonella and E. coli, that but kind of they have antibacterial activity. So after that, those compounds which provide us very good activity in the against the Salmonella and the E. coli, we then advance them for the FICI, which is the fractional inhibitory concentration. So that means that the MIC, which is the minimum inhibitory concentration, that were cross through the checkerboard method. So we shortlisted 3 main compounds, which is the cinnamon, tezpat, and the clove. And we check that which one has the good ability. And after that, we see that these 3, tezpat, clove, and cinnamon, these are all the names of the plants. They were very good in their composition to provide a good growth promoting ability, and that can combat both the organism. Actually, Salmonella and E. coli, their influence is so much in the Pakistan poultry. So this is very important to combat these kinds of bacteria, as they belongs to the Enterobacteriaceae family. And if we can combat them instead of using antibiotics, but through using some of the natural or herbal compound. So this was basically the main idea. And then after certain other kinds of testing, I have not shown here. So we directly go to the product formulation. And for this product, we have categorized them through various composition methodologies. And after that, we test them in the poultry and we see that it has a very good effect on the bursa. Bursa is an important immunological agent, immunological organ present in the poultry. So we can see that the size is very high. And also the spleen, which is also an important immunological organ, especially for providing the cytotoxic immunity. So its size is also going high. And also these all, the product formulation compound, they increases the weight in the poultry and also restoring the histopathology, especially in the spleen and in the liver and other immunological organs. So then we registered that product through the IPO, which is the International Intellectual Property Organization, and it is almost present in all of the government. And we registered that product through that. So secondly, another publication in 2023 that was actually we planned it to see that if trace elements like zinc, cadmium, copper, if they are present in the grass or in the forages, so what could they have impact on the grazing sheep due to gastrointestinal burden? So we conducted that study in two main districts, Sialkot and Multan of Pakistan. We see that in the Sialkot district, there is actually 34% burden of, or you can say the prevalence of the gastrointestinal organisms, and in Multan, almost the 32%. So we see that if copper and zinc, they are in the high amount in the forages or in the soil of these two regions, so they can easily combat the gastrointestinal parasites. But if they are lower, so if these microminerals, they are lower in the forages, then they cannot combat. So means that the intensity of parasite infection was lower and the level of trace elements in the serum was higher. So that means that this creates a very good influence or a very good correlation to combat the GI microorganisms. So in another study, which we have conducted in 2022 about the probiotics, and this probiotic contain actually the yeast Saccharomyces cerevisiae, we wanted to check their impact on the egg quality or the weight gain, and also in the immunity of two main layers, the poultry layers. One species is the Australoop and the other is the Phaeomi. So we actually designed the methodology like this. We have given these three kinds of feeds, 0.5, 1 and 1.5 gram the per kg of the feed. So we see that there is good impact when we give 1.5 gram, means increasing the dose actually increases our parameters like egg weight, half unit. Also, the yolk weight was getting high and richer in the 1.5 and also in the 1 gram. Also, the yolk ratio was very good. And the eggshell, half unit, eggshell weight, and albumin weight. So that means that if we increases the dose of the probiotic, that means that can increase the required parameters of egg and also its weight. Then another study was conducted in 2020 about the prevalence and hematological alteration. We want to see that against the hematological parasite like the anaplasma, which treatment protocol could be a good one. So we conducted these two kinds of treatment strategies, one with the antibiotic oxytetracycline and one with the enrofloxacin. And imidocarb dipropionate, which is the recommended drug against the anaplasma. So we give that drug in both kinds of group and compared them with the control group where there is no treatment was given. We see that oxytetracycline along with the imidocarb has the better result compared to the enrofloxacin only. So the parameters that can assess like the risk factors which belong to increasing the prevalence of bovine anaplasmosis, there are so many, but the impact was greatly influenced through the age. Like young one has the more infestation problem, also the cleaning time, that how much time we can clean, that can also impact, and the feeding system, floor system, hygiene measures, and the caricidal system. However, we cannot change any kind of differences through the sex, like male and female. They are not significant. Also, the herd size, large or small, they cannot have impact significantly. Also, the farming system. So, regarding the hematological impact, we can see that that this anaplasma, they have a very pronounced effect in decreasing the RBC, PCV, and HB and MCHC. These all parameters, they actually belongs to the anemia, kind of special anemia, which is the microcytic and hypochromic. So it means that anaplasma, which creates the microcytic hypochromic anemia, if we combat this organism through this treatment, oxytetra and imidocarb, that can give a fruitful results to recover the animal from this menace. Next, another study which is actually related to the goat meat, the effect of age, sex, and chilling. We disclose only the important parameter like this temperature. So there is a myth in Pakistan that milk teeth animal, they have a better meat quality compared to a very adult or two teeth animal. But actually, we want to show that it is not the milk teeth or the two teeth animal. Actually, it is the temperature, the cooking loss. So temperature means that if we delayed chilling, is that better or the rapid chilling? So importantly, we see that if after slaughtering, we directly put the meat in the refrigerator, that will be a rapid chilling, and that will not good for the tender quality of the meat. But actually, it is important that the delayed chilling, that means that if the meat, it goes to the rigor mortis first at the room temperature, and then we put it in the refrigerator, the meat quality and tenderness will be more better. Also, the sex, like male and female, they do not have pretty much important in the meat quality. However, if we see the cooking loss, then males have more cooking loss because they have maybe they have more protein content as compared to the female. So the cooking loss is also higher in case of the males. So then in China in 2018, there was another kind of study which was conducted, and it is actually related to if we do the RNA sequencing, and the procedure is that if we go through the boron supplementation. So boron is a micromineral, but we want to check that if we give that micromineral to the animal, uh, what kind of impact on this, on its immunological process can be happened. So idea was that we cannot check all the immunological parameters, so we just select the MAPK signaling. So for the boron, we actually divided three doses. Two doses mainly. One is the control group, and other is the 80 mg per liter and the 640 mg per liter. So mainly what we see that more genes were downregulated through this analysis, RNA-seq analysis, because it provides us the differential gene expression. So in the differential gene expression, a little bit summary, we can see that only 309 genes were upregulated, but more were downregulated. And similarly, in case of 640, when the dose was higher, again, more genes were downregulated. So we shortlisted the whole data in 7 profiles. On the basis of that, one profile in which all the genes are going down, and in other profile, some were first stable and then go down, some were first go down and then stable, some were go down and then increase, some were stable and then increase, some were increase and then go down, and some were increase and then stable and some are only increasing. So we can see that increasing contain only 144. So 5 profile we do not see because their number of genes are less. Although we cannot say that these are the non-significant, but for better approach, we only selected the above 3, 0, 3, and 1 profile in which a lot of genes were present. So we see that in the MAPK signaling and in the calcium signaling, there is an immunomodulation. Like see, this is the KEG pathway in which we can see that the MAPK is involved in so many areas. These green areas are actually the downstreams of all these MAC. And through the Western blotting, we can see that all the JNK arc, they are going high. And because the MAPK is actually upregulated, and this is important to see that the boron supplementation increases the immunity of the animal. So now comes to the background picture of the Indonesia. We can see that in Indonesia, although the livestock sector is growing, and yes, it has to be grown because the number of population, the population of human beings are going high. So the demand of food, especially the meat and milk, is getting day by day higher. So, but especially from ruminants and cattle population, they are actually exposing through various challenges like disease outbreak, and there is some sort of a lack of infrastructure, or the biggest or commercialized like the corporate sector farms. Also, the reproductive losses due to long calving interval. Maybe it may be due to the less reach of veterinarian at a proper place to provide the chemotherapy or the treatment, and also the low conception rate, which is only a 56%, and internationally recommended almost 70 to 80%. And then there is high calf mortality initially, and main probability of this calf mortality may be due to the diarrhea, especially the infectious diarrhea. Then those animals may have the high dependence on live cattle and beef imports. So that's why Indonesia is currently importing the meat and also the milk to meet the needs of their people. And also there is a chance of poor feed quality, and smallholder farmers are more. So they although this is important for their economy, but when we talk about the overall picture of the country, then we need actually the commercial and corporate sector to gather the animal at a certain place and then to improve their genetics so that they can be highly productive. So I really appreciate the government program, the SIVAB, and although it is partially successful, but still it is encountering through several challenges, and maybe these challenges belongs to the reproductive losses. So one possible way which we can design that, although the adult animals now they are performing, but if we increase the survivability of the calf and also increase its improvement, so the future generation of the livestock can be improved. And one possible and the advanced technique is through the fecal microbiota transfer. So what is that? Fecal microbiota means we have to select some sort of animals that can act as the donor, and they have a very solid amount of microbiota within their rumen and in the intestine. So we can collect them and then transfer it to those animals that are diseased. So instead of using antibiotics or chemotherapeutic agents, we can give them some natural kinds of bacteria that can help them to resuscitate their gut and the environment of the gut, so they can be more healthy, more productive, and they are the farmer friendly and the healthy. So they cannot, if we provide them the chemotherapy or the antibiotics, so definitely it's a burden of chemicals on those animals. So once, although they becomes healthy, but the residues of these drugs can be deposited in the milk and the meat. So ultimately, these becomes the part of the humans, and human can be resistant to several kinds of antibiotics. So we need a safest strategy, although this strategy is right now advancing and people are working more and more for its optimization. So there is a rough estimate of current scenario of Indonesia, like the estimate increase from 2024 to 25 for the milk is almost 9% increase, and it is day by day increasing. So the demand of milk is almost 3 to 4 million ton, and the production is almost 0.6 million ton. So it means there is a gap. Similarly, for the beef production, this is the present estimate in tons, but the estimate we required is almost 7,40,000, which is 0.74 million ton, and almost per year 2.3 kg is increasing. So meat supply, although better than the milk supply, 60% of the demand, but still 40% is lacking. And also the milk consumption is increasing, like currently in the last year 24 16.3, but now the demand per year is rising from 25 to 30s, and same is almost the case with the beef, although with low increasing the demand. So there is a comparison in 2010 and 2021 about the domestic production, which was 43%, and now even after 11 years, there is a 2% increase in the demand. And the import was 17%, which is now 33%, almost the double. And same with the domestic production, like 40%, and it is a decrease, maybe due to high import, or maybe due to the disease presence. So that means there is a requirement, there is a demand to improve the livestock sector. So animal fecal microbiota metagenomics is important, because profiling that what kind of microbiota is present is highly important. If we know that, because in the rumen and in the intestine, there is a mixture of bacteria. There are the pathogenic bacteria and also the fruitful bacteria. So their profiling is important. Like here in the A section, we can see that the pathogenic bacteria. In the C section, we can see that the fruitful bacteria are present. So that is why what these bacteria are doing actually, these fruitful bacteria are providing a competitive environment with the pathogenic bacteria. They're also providing SCFA, which are the short chain fatty acids that are necessary in the ruminant animals and the livestock to provide them the glucose, and also provide them those kinds of fatty acid, which ultimately becomes the portion of fat within the milk and the meat. So they also increase the immunity, and the mechanism is provided here. So basic objectives are, we should have to know the taxonomic composition of these bacteria and what kind of diversity they have, what is their functionality, how much they are stable or the persistent, and what kind of pathogens they can be present and provide a competitive environment to these fruitful bacteria. So going forward for the technique, so here is a short summary, and for with each one I have provided a YouTube link so that people can understand that how they can go through the whole procedure. So first we have to get the fecal collection, and there is a special technique through which we have to collect them and we have to preserve them, because the main environment within the rumen is the anaerobic, and it is highly important to preserve these bacteria because they are coming from the anaerobic environment. And once we collect the feces and expose them through the environment, there is a chance of oxygen exposure and they can be killed. So this is important for their preservation. And secondly, we have to dry the fecals and then weight them before using. And then the filtration to get through only the bacterial or transferring community and not the other residues or other feed ingredient that could be a part of the feces, they must be removed. And there are certain storage material like NAP, Omnigen, RNA letter, in which we can preserve them, these fecal microbiota for a longer time at minus 80 degrees C. Then the results of this kit is highly suitable, and This kit, although there are several kits in the market available, but this one, QIAGEN, it is highly reliable and provide more better results. So in this way, this picture shows that there are several donors, and when after collecting their microbiota and extracting their DNA through using this kit, so we can see several kinds of bands. So one band is actually a mixture of DNA from several microbiota, and this 1, 2, 3, 4 up to 7 represents various samples. So then we check the quality of the DNA. So after that, there are two approaches for the library preparation. So first is the targeted, and the second one is the shotgun approach. Targeted means we know our target. So it's an amplicon based technique. So targeted means they are from the 16S or the ITS region. So what is 16S? It's actually a conserved region within the bacteria. So this has been explored that from V1 to V9. But Illumina mostly detect through V3 and V4, although V1 to V9 several primers are available. But because Illumina is pioneer in this sequencing and highly reliable, so they mostly consider this V3, V4 for the metagenomic analysis. And this one is their strategy to use this 16S primer, and at a time we can conduct 384 samples, and this link provides that we can also do a short training course for this 16S sequencing. So they have designed these two primers. We can see that these are a very long nucleotide sequence primers. So this is necessary for their library preparation, although there is one step before the library preparation, because our DNA is a mixture of all kinds of genomes that are obtained from the microbiota. So that means that in the library preparation, we have to cut or fragment those to get the shorter reads. But actually, this is the beauty of 16S, because we already know the primers and already know our targeted approach, that which one we have to amplify. So for this purpose, we don't need to fragmentation. So we directly go for the PCR as the primers are available. And this is the strategy that we join these primers and amplify them. So with these primers, there is a sequence index 1 and index 2. So on the basis of these indexes, when we send our sample to the company for sequencing so that they can provide us that what we have obtained, we tell them about the index which we have used. So this one is the ITS region. It is almost resembled with the 16S, but the difference is that 16S is for the bacteria which are present in the microbiota, but the ITS is for the fungi which is also present in those microbiota. So for this case, there are two regions which has been selected, ITS1 and ITS2. This ITS is basically the spacer region, internal transcribed spacer region. These are also the ribosomal RNA genes, but present in the fungi, not in the bacteria. So here we have to give a little bit difference with the two-step PCR, not the one-step PCR as we have previously done in the bacteria. So in the first step, just like the 16S, the long primers are available, forward and reverse, and we just simply give the PCR. So this is the step 1. But in the step 2, because several fungi are available, so their ITS 1 and 2 regions are different. So that's why they provide us, I mean the Illumina, in their kit, they provide us 8 forward primers and the 7 reverse primers. So with the help of them, we amplify the library in the second step. So all these primers, like the 7 forward and 8 forward and 7 reverse, they have different indexes. So indexes mean major sequence is same, but a small sequence will be different. So this different small sequence that may be from 5 to 7 nucleotides, This is actually also present with the company that will go for the sequencing, and they will identify that, okay, if they have used the index 1, so they will also run their sequencing through the index 1. And if we have used the index 2 or 3 or 4, in the same way, they will go for the sequencing. And in this way, they can detect our reads and provide us the data in the faster file. So this one has been presented in a summarized form, that this library portion, the finally, in which our adopter sequence and our primer sequence is attached with our read going through the sequencing. So that is why we got two large peaks in case of fungal metagenomics, because there is one region ITS1 and the second one is ITS2. So this picture, the sequencing company will also send us, and we compare it that if we are getting these two solid and long peaks, sharp peaks, this means that our library quality is good and we can go for the further analysis. But in case of, now come to the strategy of the second, which is the shotgun sequencing flow. This is a non-targeted approach, and I mean to say that it is more costly, but it is more effective and more beneficial, because in the targeted approach, we know which kind of bacteria we are going for detecting and which kind of fungi we are going for detecting. But in the shotgun sequencing, we are actually gathering throughout the genome that are obtained from the metagenomics. So this also provides us those kinds of bacteria which we don't know. We have to match them in the database, and maybe we can get and identify several new bacteria. For example, in one environment of the Indonesia, they are offering them a very good quality of the feed. So the microbiota of those species of cattle is quite different from those animals which are present or habitated by a farmer, which cannot be able to provide them a good quality of feed. So definitely the difference in the animal health and the animal texture, animal composture, that is also based on the diet, and this diet directly impact on the microbiota. So shotgun sequencing actually provides you a broader picture, a larger picture, and give you several fruitful results. So for this purpose, definitely, if our genome is unknown, we don't know, we gather it from the several species of bacteria, fungi, and the protozoa. So first of all, we have to fragment it, and then we will go for the library preparation. I mean, for the adapter ligation and then indexing, and then send it for the sequencing. So there is a brief summary that, for example, this is a whole genomic DNA obtained from several species. And after that, we will cut it through several restriction enzymes, which means we will fragment them. So we will get so many fragments and then ligate them with several adapters and then amplify them through the PCR, and then we will send it to the company. So in the metagenomics, the shotgun approaches, several kinds of kits are available which are provided by the Illumina, but the better one which is now in use is NextEra XTDNA library. The second one is in high use. So basically, they're providing, this one is more advanced and this one is the previous one. So only difference is in their utilization. For example, in the M tegmentation means fragmentation and applying the adapter at the same step. So they provide magnetic beads, and on this magnetic beads, restriction enzymes are already attached at equal spaces. So they capture the genomic DNA, cut it in the equal parts, and then join them. And after that, adapter are ligated. So we just have to give a PCR to amplify the library. So that is why we only get a single peak, because cutting is at the similar site. But in this case, which is now in use, they provide two cutting restriction enzymes. So they cut it at several places. So the read lengths are not same. Some are short, some are broader. So we will get two kinds of peaks. The narrower peak represents the shorter reads and the broader represents the broader reads. So for the when we send these results to the sequencing company for sequence. They will provide us the FASTA file after trimming and after quality checking. So they provide us. So we will go through the bioinformatic analysis. So I have given this training course here that first we will subject it our all reads. So these reads will be in these reads will be in the FASTA format in the nucleotide sequence. So first of all, we have to convert them into the amino acid sequence so that we can annotate them functionally. So we use several databases like the MetaGeneMark. Then we subjected them into the Interpro to scan whether these reads they have some sorts of genes, and if there are genes, whether they are functional or the hypothetical, and even if they are functional, what kind of function they are providing, what kind of biological processes they are taking part, or the molecular processes in which they are taking part. So the limitation of Interpro is that at a time it takes almost 100 protein sequences only. So if the gene numbers are large and high, so then we have to make several sheets to subject them into the Interpro scan. Then for the COG number detection, we will use the eggnog because COG number is also helpful in the functional annotation and identification. And then for checking which kind of bacteria are present to get into the broader database SkyBlast. So one by one, we will add the sequence and get the bacteria. So then we can classify that which kind of bacteria are present in these kinds of sequences. For example, if we are getting 50,000 sequencing and out of 50,000 sequences, 10,000 are belong to one kind of bacteria, 5,000 from others. So in this way, we can categorize several bacteria, also several fungi, also several other protozoa. So we can check that which kind of taxonomy is present. Also, this blast will also be helpful that if we are getting several kinds of bacteria or the fungi, whether they are pathogenic or the beneficial, because now the databases are so broad that we can easily detect that they could be from the pathogenic environment or from the beneficial environment. So because these bacteria and fungi, they are also present within the GIT environment. So if we are giving some sorts of antibiotics, or if, for example, in the farm community where the animal are present, if in the forages or like in the water, there are some residues of the antibiotics and animal are taking feed or diet, so they're also getting those antibiotics from the environment. So although we have not injected the antibiotic, but they are getting from the environment. That means those bacteria which are present in the stomach, they can also be encountered with those antibiotics and they can express certain kinds of genes. So although our metagenomics is beneficial, but the bacteria contain the genes for those antibiotics. For this purpose, we will subject our whole data into the CARD database, and CARD will tell us that which kind of resistant genes or markers are present, and whether the identified bacteria, they are sensitive or not sensitive. So in this way, we can categorize that whether our metagenome is good, filled with so many beneficial bacteria, but there is a high chance that even in the presence of these beneficial bacteria, certain antibiotic resistant markers or genes could be present. So another way is that instead of doing this rigorous methodology, although this is important for manual checking one by one, we can also subject them into a KEGG secondary data environment, which is the ghost koala. We will subject our throughout FASTA sequences by uploading them and can get this picture in a pie graph way. So there is one another advanced technique, although it is not commonly used, but for the interest of the researcher, I'm describing that there is a strategy. For example, in the Mycobacterium tuberculosis, I'm just giving a shorter background. CRISPR-Cas9, it is a highly common. But in the Mycobacterium tuberculosis, instead of CRISPR-Cas9, there is a presence of CRISPR-Cas10. So because what is Cas9 is doing? It is actually doing the genome editing. So what we have done, that if we reprogram Cas10 of the Mycobacterium tuberculosis, so instead of giving any exogenous Cas9, its internal Cas10 will be helpful in the genome editing of that bacteria. So that bacteria, because it is conducive for the Mycobacterium tuberculosis environment, so it can be optimized very easily. So in the same way, what we are planning actually, that if we have seen certain kinds of beneficial microorganism, and suppose these microorganism within the microbiota, if they contain several non-beneficial genes like the antibiotic resistant genes, and we want that microbiota to transfer into the recipient, but we don't want that those antibiotic resistant markers from the donor should be transferred to the recipient, then we have to edit their genome. And for this purpose, we can go through several strategies. So one possible way is that we use the virus, like adeno-associated virus or the lentivirus, and these are the pseudoviruses, not the actual or disease-causing viruses. And the second strategy is that we just take those beneficial microorganisms, and just like Mycobacterium tuberculosis Cas, we can reprogram their Cas and edit their genome. So in this way, those microbiota will be our edited or genome edited microbiota, and that will be our patent. We can store them for a longer time. So whenever we want to give them to the animal, so we can say that this is our genome reprogrammed or genetically edited probiotics that can be highly helpful for the further for the recipient animals. So lastly, this review, I have given its reference at the end. So they're also planning like that. These are, for example, the probiotic bacteria. There is a list, and these are the plasmids. So if we can use these plasmids, for example, I want to work on the L. crispatus, the first one. So I will use this plasmid, PTRKH2. So I will induce those genes in that plasmid that are already present in L. crispatus, but I want to express them more. So in the plasmid, I will insert those genes that are more beneficial. For example, they like these are certain kinds of enzymes gene that can degrade the fat to provide the energy more, or they can degrade the carbohydrate and provide instant source of energy. So in this way, our L. crispatus will not be a simple L. crispatus, but it will become an overexpressed L. crispatus. Similarly, the same plasmid, if I induce Cas9 gene, and with the Cas9 gene, I also incorporate a guide RNA for the gene, like oxytetracycline or like the enrofloxacin or against any other antibiotic. So my purpose is that if I give this plasmid to the L. crispatus, so its CRISPR-Cas9 and guide RNA will go towards those genes in the crispatus which are expressed for the oxytetracycline. So it will cut that gene and remove it. So in this way, this L. crispatus will be transferred to the animal, and now this L. crispatus will no more contain any gene that can provide resistance against the oxytetracycline. So that provision of a probiotic means that it is safely used in the recipient without the inculcation of any kind of antibiotic resistant markers, and that animal will become more health beneficial effects. So thank you very much. These are the references for the several data I have presented. Thank you very much. And if have any question, please ask.
Thank you, Dr. Ahsan, for the enlightening presentation. I think it is very interesting for researcher or people who have interest in metagenomic, genome editing, and then someone who want to produce feed editors to want to evaluate the productivity and health in animal. So it is very interesting from your previous study, how you develop herbal product to determine the antibacterial potential to poultry. And one thing I might highlight in your presentation is about fecal microbial transfer. That also my ongoing study. It is amazing how microbial communities in phases can be utilized to support animal health and disease management. And also your explanation how to provide from an underlying mechanism to practical applications in genome editing also. We truly appreciate your contribution and your presentation, Dr. Ahsan. And then there are some questions from participants. First from Mr. Tanda Kain Jaitan, thank you for your interesting Pak Tanda. Dr. Ahsan, maybe we directly jump to the question and answer session.
Okay.
Okay. First question is very interesting when you add probiotic Saccharomyces in layered diet. What level of protein and energy you set for the formulation? And also the second question from Dr. Tanda. He agreed that chilling made goat meat more tender. Though some people said no need to go further with aging in goat or sheep meat. What do you think? Maybe two question first. The time is yours.
Thank you very much. So actually, we use the commercial diet, and because we were doing in the layers. So in the layer, normally the feed number, when we provide the feed to the animals, like in the broiler, it is commonly called 22 number feed, and in the layers almost 18 or 20 number feed. So I'm not so sure that through these numbers what kind of proteins they are giving, but I think that in the layers the recommended one is almost 15 or 16% or like 18%. So so we just use the commercial diet and it is already optimized. Our purpose is that we just want to see that along with this commercial diet, if we provide certain kind of probiotic, and by increasing the probiotic dose, what kind of beneficial effects we can get. So this is what we can just do in that. And secondly, for the question about the chilling effect, thank you that you are agree with that. So yes, you are very much right that if we take the meat of the animal, and because it is a myth, and directly people said that if we place the meat outside after slaughtering, there is a chance of infection or contamination, or the meat quality will be deteriorated due to temperature, or due to the environment, or due to flies or certain kinds of insects. But the point is that if we directly put them into the chilling environment, so this will shorten the length on the fiber length of the meat. And if it is in the contracted and shortened form, definitely the tender and the quality will be deteriorated. Also, within the meat cells, the fat molecules are present. So these fat molecules will not be utilized for the cooking purpose if we directly chill them. So if we just place them outside, this fat will also be distributed, or at least gone towards the membranes of the cells, and meat will a little bit in the expanded form, and its fiber will not be in highly compacted or in the contracted form. So After that, technically we say that the rigor mortis. So when rigor mortis will appear, maybe after 45 minutes, or some says that within first 2 hours of the slaughtering, then we can place them in the chilling. And also recommended is the delayed chilling, not directly in a high freezing environment, first like in a gradual or step by step, like through the refrigeration, then going towards the deep freezing if required for a longer use. Thank you.
Thank you, Dr. Ahsan. Dr. Tanda, do you have any comments or suggestions, please?
Thank you. I just want to have a further comment from you regarding to the aging. Some people say when you slaughter goat or sheep, you don't need to go for aging. What you comment on it? Thank you.
Can you repeat it, please? Sorry, there is disturbance in the voice.
Okay. I just want to ask you further on the. Okay, I agree about the chilling, but then some people mention that we don't need to go further with aging to get a tender meat in goat and sheep. So, what do you reckon on it? Thank you.
Thank you for the question. You're right that we don't need to go with the aging, but here I have also written here. It is a common myth in Pakistan. I don't know why, but it is persistent myth in the local community, local population. They said that the milk teeth animal, its meat quality is more good, and we have to consume that. And they think that maybe the meat is soft, easily cooked, so they think that its digestibility is more better as compared to a very adult or a very two teeth or more teeth animal, for which cooking is a little bit difficult. So we actually just want to overcome that myth through applying a shearing force. So, and we want to check that whether it is a real thing, that the myth is quite be translated into the reality, or it's just a myth. So it's a misconception. Although the animal is a milk teeth or two teeth, so age did not have any kind of significant effect. And I have also written it here in the first line of the conclusion that age do not has effect, and I agree with you. Okay.
Okay, Dr. Tanda, do you have any suggestion or comment?
Nothing. That's enough for me. Thank you.
Thank you.
Thank you, Dr. Tanda. Maybe one more question for Dr. Tanda. He asked about, do you have any comparison study in fecal microbiota transfer that work to some extent?
Comparative study.
Comparison study in fecal microbiota transfer. Maybe Dr. Tanda, maybe the comparison study of fecal microbiota transfer and also with antibacterial study. Is it true, Dr. Ahsan and Dr. Tanda? Can you explain again about your last
I just want to
question?
get some. I just want to get some information on the comparison study. When you mention about the fecal or feces microbiota transfer, and it's improved many things in the gut. So, do you have any kind of a study that been done on it, or something that a published paper that mentioned that's what you put in that criteria? That's what I'm just curious, want to know what is the effect when you do that. Thank you.
Thank you. Dr. Tanda, thank you for your, again, a very precious question. Actually, highly relevant answer to your question is that recently with the BRIN host researcher, a call was open in the Higher Education Commission of Pakistan, and we have recently submitted a proposal in that. It was actually, just I will disclose a brief point of that. we want to fortify that fecal microbiota by adding certain substances. So our aim is that fecal microbiota usually people use, but if we do its fortification, what kind of beneficial effects we can get? So let's see that right now we don't have study, but proposal is submitted, and if that proposal is accepted, then definitely we will go for its full length project submission and go for the further procedure. I hope you got the answer.
Many thanks. I hope you get your proposal get done through. Thank you.
Thank you. Thank you.
Thank you, Dr. Asan and Dr. Tanda. We will move to the next question from Mr. Anshar. Okay. From the projects Dr. Naim has conducted, which feed treatments, additives, or supplements have shown the best effects in improving livestock production and health, particularly from the metanogenic and immunological perspective.
Thank you for this question. Thank you for this question. I think the question is from actually the question belongs to our this project. So this was conducted on the broilers. So in the broilers, we give them commercial feed number 22, and we take the animals right from day 1, the short chicks, up to day 35. So along with the feed, we provide them our herbal product. And for the herbal product, we have actually given 2 doses, 1 ml and the 3 ml in the per liter water. So this herbal product is actually a mixture of 3 compounds. Plus, we have also added 2 more things. And now because it is a patent, so I can disclose it here that we have added selenium, vitamin E and vitamin C. And along with that, we have used the paraben preservatives. So in a special quantity, we have mixed them. So this quantity we can obtain through the checkerboard method, FICI. So it provides that how we have to divide the plants, and then in what proportion we can use the other supplementary material. So daily we provide them the diet like the group for which we have given 1 ml and the other group with the 3 ml. We have provided them these diets daily right from day 1 up to day 35, and side by side, so we will check we were checking their weights, and time to time, like from the control group, we slaughter an animal to check the immune response on their bursa and also the spleen. So one data is also, I do not show here, it is the ELISA results of certain cytokines. So we also check those results. And after the age of 35, we slaughter all the animals and then conducted the histopathology and all other things. So in this way, we see that our herbal product works very good. So the main aim was because in the poultry sector, there is a lavish use of antibiotics. So we want to see that if there is any approach through which we can combat the antibiotic effects. And although we cannot completely subside antibiotics because certain infectious agent cannot be killed through the herbal medication. So our aim was that along with the antibiotics, by decreasing their utilization and increasing or complementary utilization of the herbal drug. So it's a mixture. So by complementing the antibiotic with the herbal drug. So in this way, we cannot thoroughly remove the antibiotic, but at least we can reduce the amount or the dose to get our fruitful or beneficial results.
Okay, thank you, Dr. Arsan. Dr. Ainsyah, do you have any comments or recommendations?
Okay, thank you, Mbak Ezi.
Dr. Ainsyah? Okay, maybe we will continue to the next question. For the next question from Dr. Retno. Thank you very much for the information, Dr. Muhammad Asanaim. Research in metagenomics must be very extensive. Is there any in any minimal sample, or maybe just one head of animal is enough to represent the treatment? Thank you very much.
Thanks for the question. Yes, I think that as I described that there are two approaches for metagenomics. One is the shotgun, and the other is the targeted or the amplicon based. So I think that donor selection is the key step for metagenomics. So if the cost is high or a project do not allow to consume more money, so my suggestion is that emphasize more and more on the donor selection, like to check its health status, body scoring, almost for the 6 months, to check it do not get any kind of disease condition, especially the diarrhea from non-infectious or from the infectious causes, no temperature. So the animal should be healthy from the last 6 months, properly vaccinated according to your environment, and also check it through the rapid kit method for certain diseases, and also through the ELISA and the PCR. And after that, when you select and you are very much confident that this animal is now our donor, plus check its body scoring condition. And one more thing, check its fecal scoring. Fecal scoring is also available through the internet. We can easily get that how to score the feces. So when you have the background knowledge about all these things, it means that now your donor is final. One donor is enough. We can take samples time to time from that donor, and we can preserve them. Now the important point is that which approach we can use for our further purpose. So I think that instead of going towards the shotgun, because it requires more expertise, more expenses, its data analysis is more rigorous, and certain unknown bacteria we can identify. So it explore more channels. Although for the research point of view, it is very good, but for the financial point, I think that targeted sequences is important. And in this targeted sequencing, we can target one or two bacteria. For example, if we select that bifidobacterium, it's also a very beneficial bacteria, or like we can say that lactobacterium or any other kind of bacteria, we can target one or two bacteria or maybe their species. So in this way, we do not go for 16S sequencing for all kinds of bacteria, even in the targeted metagenomics. So we will get only 16S sequencing of those particular bacteria. And if they are present, we can isolate them through the biochemical culturing and the PCR approach. We can isolate those bacteria and then only characterize those bacteria by simply doing the qPCR for checking is there any presence of the antibiotic resistant genes. And if these genes are not present, it means that our bacteria is not only beneficial, but also free from the resistant genes markers. So now we can culture that bacteria, increase its level, and then dry it, or even we can lyophilize it in a dried form for using further purpose. Like we can make the capsule of this dried powder of bacteria to transfer it into the recipient animals. So at least our studies can go further. Secondly, you can also use your bacteria with simple genetic approaches, like I have discussed it earlier, by simply using cloning method. For example, I tell you one thing. This licheniformis, for example, if we use this bacteria and we are using this plasmid, we can just select one or two beneficial genes. And what are those beneficial genes? We can easily get its genes from the KEGG pathway analysis, or even from the certain databases. For example, energy biomarkers. We want to see that if this bacteria contain an enzyme that can degrade the lignin easily, or like it can degrade the carbohydrate or cellulose very easily. So we can just amplify that gene and clone them into this plasmid vector, and then put that vector into this bacteria. So now this bacteria contain two those genes, one from the plasmid and other already present in its genome. So we called it this bacteria now as overexpressive bacteria. So that bacteria will now overexpressive that gene, means produce more enzymes. So you can check the effect first experimentally and then produce it as a commercially. So we don't need to go for shotgun sequencing if we have financial problem, but first we can start by narrowing our strategy through the 16S sequencing. Thank you. I hope you can understand.
Thank you, Dr. Ahsan. Dr. Retno, do you have any comments for the explanation from Dr. Ahsan?
Thank you very much for the explanation. Thank you.
Thank you.
Okay, thank you, Dr. Retno. We move to the next question from Dr. Diki Pamungkas. Dr. Ahsan, is there any correlation between the targeted organ by supplementation and fecal microbiota metagenomic analysis in terms of enhancing immunity in poultry or ruminant?
Enhancing immunity in poultry
Enhancing immunity in poultry and or ruminant.
Okay, through which metagenomic approach?
A bit, okay, I explain again.
Yeah.
Is there any correlation between the targeted organ by supplementation and fecal microbiota metagenomic analysis in terms of enhancing immunity in poultry or ruminant?
Oh. So there is a question, and I think this question is highly related to the brainstorming. The question is that, is there any correlation in that the targeted metagenomics and its correlation with the diet that can induce the immunity? Yes. For this one, we get to know some sort of background knowledge. For example, as I'm explaining before, so how in the poultry and livestock immunity increase? Actually, if we categorize the immunity, so there are two kinds. One is related to the antibodies, like the humoral immunity, and the second belongs to the T cell immunity, like the CD4, CD8, these kinds of immunity. So the question is that if we are increasing the immunity, like outside from the diet source, through which procedure we are increasing immunity, like from the mineral source or the vitamin source, or through which way? So we can take an example just to understand. I'm not sure how much this example is relevant, but to understand the query, it is highly important. For example, there is a diet, and that diet contain high content of fibrous agents, but also we get to know that this diet also contain several vitamins and minerals, but these vitamins and minerals are entangled in the fibrous portion. So definitely there is a limitation within the animal gut that it can consume fibers at a certain level, but not at a higher level. So at a certain level, when they are consuming the fiber, so at that level, they are getting the vitamins and like this. So this could be a case that because this kind of diet mostly cheaper, it's like a roughages type of diet. So definitely when animal consume it, although animals are surviving but not so much productive, so how we can increase the potential of that animal to digest it more thoroughly and get more and more vitamins and minerals, and in this way its level of immunity will be high. So for this approach, yes, we can use the targeted metagenomics. For example, instead of using targeted metagenomics for all kinds of bacteria or fungi, first we have to see the literature to shortlist only one or two bacteria or the fungi that that has the potential to degrade that lignin on fibrous kind of diet to digest it very well. So after shortlisting that, we can go on through its metagenomics, 16S metagenomics, by collecting the fecal microbiota from a donor. So by using that 16S, first we can identify that yes, this animal, that this bacteria is present in our donor or not. So if that one is present in that donor, so we can get that bacteria by purifying it from the microbiota. So after that, we can, just like previously I told, uh, either through the genome editing technique, but that technique is costly. So simple cloning is good one. We can simply amplify that relevant gene which is present in that bacteria that is helpful in digestion of the fibrous material. So we can simply amplify by designing the PCR, amplify it, put it into the plasmid, and then that plasmid is incorporated within that bacterium. So now if we use that bacteria again, so instead of using the whole clump of fecal microbiota, we specifically just use that quantity of bacteria in a sense of an overexpressed probiotic to the animal in along with its diet. So the same refugees material we offer, and we see that now the digestion is that improved or not, and I'm sure that it will be improved because now the level of enzyme, level of gene, they are high, so they will release more vitamins, more minerals. So if we are not using anything else on the same situation with the same environment, just by utilizing a technology, we can check that whether by consuming more vitamins and more minerals, the immunity level will be high. But I believe that this is just the application of this technique, but definitely we have to improve the diet. We have to give certain other material. But if we are just focusing that we are using just this kind of diet, and to the farmer community level, which cannot be economically more grown, we can offer them a certain cheapest material to use them in their limited resources to improve the immunity of their animal. Then yes, this could be probable and optimistic technique in such a way for utilization. Thank you.
Thank you, Dr. Ahsan, for your explanation. Dr. Diki Pamungkas, do you have any comments for that explanation?
Okay, thank you.
Any
Thank
more
you.
questions? Thank you.
Thank you.
Okay, thank you, Dr. Diki. And next, the question from Dr. Tri Puji Priyatno. Dr. Naim, in the last slide, we saw several genome-edited probiotic microbes. Have probiotics containing these microbes been commercialized, or are they still limited to research? Thank you.
Thank you for the promising question. This figures and this one, I have taken the data of Liu et al. Paper. It's a review paper. This one I have given. It's reference. So actually, in this review paper, they have suggested. It's a promising review paper because their suggestion is that to improve the probiotics by utilization of this plasmid. So up till now, people are working on these projects to improve the quality and level of probiotics, but recent data is still scanty. And one thing is highly important, that even someone is working on any kind of plasmid and any kind of its relevant bacteria, still the utilization in their environment and in other environment will be different. For example, one person is using them in China, so the diet, the quality, the animal genetics is totally different. And the other person is utilizing in a certain region of Indonesia, so its utilization is totally different because the genetic composition of the animal and the environmental circumstances are totally different. So I believe that we should focus on this table and get to know the easily available plasmid. And that is very important, that if we have a library of the plasmid and the bacteria, and definitely BRIN will have that. So by manipulating those bacterial species with their relevant plasmid, we can generate certain genetically modified bacteria and then test them time to time for certain purposes. And in this way, we do not have to give any additional kind of diet, but the same diet can be used efficiently by just manipulating the bacteria. Thank you.
Thank you, Dr. Ahsan. Dr. Tripuji, do you have any comments or recommendations?
Okay, thank you very much, Mas Dia.
Thank you, Dr. Puji.
Thank you.
Tripuji. The next question may be, Bapak Ibu silahkan memberikan pertanyaan menggunakan fitur raise hand atau chat melalui chat box. Meanwhile, Dr. Ahsan, while waiting for the next question, I want to ask some question for you.
Okay.
I curious about FMT, fecal microbial transfer. In your experience, what are the key factors that determine the success of FMT in livestock, especially in ruminants? And the second one, what are the main challenges in applying FMT in field conditions, especially in large scale farms? Thank you.
So there is an important question. For the FMT, it's a novel technique. People are still using and utilizing it with very efficiency. But the main challenge, the main important point is its reservoir or the source. So I believe that a healthy donor, that is highly important. And especially that donor must belong to the same environment from where we are giving the same recipient. Means the donor and recipient should have the same environment. In this way, their metagenome will be closely resembled, or at least their metagenome can be easily transferred from one species to other species. If we just imagine it like a blood transfusion. So what we do during the organ transplant or the blood transfusion, we first screen the blood or the organ. And for this purpose, we first check the whole animal, whole individual. As an individual, we screen them from the diseases, also their blood, by testing them. And everyone knows that for the blood transfer, we do the minor and major cross matching. So, in the same way, if the animal for which we are selecting as a donor, it belongs to some other environmental situation and the recipient from some other. So there is a long gap in their metagenome. So it's better that your choice and selection for the donor is highly important. Secondly, if we have tested the donor, but there is some improper testing, so there may be some subclinical situation, subclinical disease situation that could be present in the donor. Like for example, we tested the donor for certain diseases, but we do not tested its blood for the hemoparasites or like for the gastrointestinal parasites. So there are some gastrointestinal parasites that are hidden and residing in the animal for a longer time, and they are not producing any clinical signs, but they are present subclinically. Similarly, certain animals, like for example, I tell you the situation in Pakistan, in the Narowal district, in which one campus of my university is present, we do a surveillance, we do a prevalence study. We see that any animal which is coming to that environment, the environment is humid. So we see that almost all animals, they are healthy, but by doing their blood smear analysis, we get anaplasma or like babesia or thaleria or sometimes 2 or 3 hemoparasites together. So I mean to say that donor we select, but we cannot select the donor just on the apparent or physical conditions because we are getting sample inside from the donor. So donor must be healthy both subclinically and clinically. Secondly, antibiotic resistance screening is also important. For example, if we get the species of bacteria from the metagenome and we consider that, okay, we can transfer it, but we have not screened it from the antibiotic point of view. So that metagenome, it is possible that it contain certain genes related to antibiotics. And if we as such transfer it into the recipient, so this could be harmful for the next animal. Maybe our recipient is clean from the antibiotic and it is already respond to certain antibiotic, but after uh, taking the sample from the donor and transferring it into the recipient, the recipient is actually now resistant and it is a kind of acquired resistance. So due to acquiring that resistance, now further antibiotic therapy will not be suitable for the recipient animal. If in case we have to do that antibiotic therapy, we will see that previously it is responding very well, but now it is not. So these challenges could possibly be happen. And this is also important that if we are not handling the metagenome properly, so it is possible that certain viruses like rotaviruses, they can be transferred from the donor to the recipient, and recipient instead of treating it get more sick. So the situation could be severe. So that's why I must to say that challenges are there. So we must be very conscious and very rigorously screen our donor while transferring the content into the recipient. Thank you.
Thank you, Dr. Ahsan. What I got from your point is we have to do some screening in fecal composition of microbial pathogen. It's the primary is rotavirus. Okay, and then the screening of antibiotic use in
And also the
dog.
subclinical screening.
And also subclinical
Screening.
This is very important for the donor.
Exactly.
Actually, what I think before I deeply learn about fecal microbial transfer, what I think is when we do the transfer, it is the fecal or the extract of the fecal.
Okay, another highly important question that what we are transferring, the faces or the content. Yeah. But, in human side, this strategy is also applied. So what they are doing in a three way. First way is a raw microbiota transferring. So what is raw means that if we are satisfied from our donor, everything is okay, clean, and also our recipient is sick, but not so much sick, means its immunity level is okay, but temporarily it is facing some problems like diarrhea or the fever. So what we can do that there is strategy of raw transferring. What we do in that raw transferring, that we get the faces and we just filter them. And for this purpose, we simply use the blender, put the feces material in that. But before that, we must know that what is the fecal score. Mostly fecal score, which is non-diarrheal, a solid form. That one must be obtained from the donor animal. Put it in the blender, add some normal saline, and just give a sphere run. So this will be blended in that. And after that, we just have to filter it. Not through the filter paper, but through the filter material, uh, just like a muslin cloth. So we can just filter it by removing the feed particles or like the straw or the undigested material. So remaining material now contain feces and the microbiota. And we can say that it's a raw form of fecal microbiota. So now we can also use that one. But this is the case when we are quite and thoroughly satisfied from the donor by testing all of things. So for this raw transfer, what we have a strategy, we can choose two routes. One route is through the rectum. Yes, from the rectum, we can take that fecal material into the rectum of the recipient animal. And second strategy is that we can use gastric tube, which is called NG tube. So we can pass this tube from the nasal cavity of the animal directly into the rumen. So from that, we can sip that from the NG tube directly into the rumen. So this strategy is also okay. It's a raw strategy, but this is effective. The secondly is to purify the bacteria, that only the content uh, of the bacterial population. But for this purpose, we have to take the fecal microbiota into the lab, and then there we have to characterize them through biochemical method, through PCR, and then to grow them on the culture to characterize that which one is present or which bacterial colonies are present. Then we can gather them, either one, either two, or either so many colonies of different beneficial bacteria. And then in the form of CFU, like in the we have seen the commercial probiotics, they have written that it contains 1 billion CFU or like this much quantity of CFU. So in this way, this is another strategy, but it's a purified strategy and more costly strategy. And the third strategy is that instead of using this purified strategy, we can just take one or two species, genetically modified them, but this will be our specific or targeted approach to target only one or two bacteria, genetically modified them, and for a specific purpose, we can transfer them from the donor to the recipient animal. So in this way, our strategies are varied according to our goals. Thank you. I hope you get the point.
Okay, thank you, Dr. Hasan. I got the point that there are three ways:
We will.
from rectum, and the nasogastric tube,
And
and also purified bacteria.
purified form. And the third one is the genetically modified.
Okay, thank you so much, Dr. Hasan. Maybe there is no question anymore for the participants.
Okay.
My question is the last question in this session. Bapak Ibu, at first, thank you very much for Dr. Hasan for your insightful and comprehensive presentation. We truly appreciate the knowledge and perspectives you have shared with us today. Terima kasih juga kepada Bapak Ibu peserta yang sudah berpartisipasi aktif dan telah bersedia ikut mengikuti diskusi hari ini. Semoga webinar ini dapat memberikan wawasan baru dan bermanfaat bagi pengembangan penelitian dan praktik kesehatan ternak di masa depan. Selanjutnya saya kembalikan kepada Bu Yeti. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih kepada Ibu Ei yang telah memandu sesi paparan dan diskusi. Thank you very much to Dr. Muhammad Hasan Naim for
Thank you.
interesting presentation.
Thank
Baik
you.
Bapak Ibu, alhamdulillah kita telah sampai di penghujung acara. Terima kasih kepada seluruh peserta webinar yang telah menyimak dari awal hingga akhir acara. Kami mengingatkan kembali kepada Bapak Ibu untuk mengisi presensi yang sudah di-share oleh panitia di kolom chat. Dan saya Yeti Listriani mewakili panitia yang bertugas. Mohon maaf apabila ada kesalahan dan kekurangan selama acara berlangsung. Izinkan saya menutup acara dengan sebuah pantun. Metagenomik sampai FMC. Banyak ilmu kita dapati. Terima kasih sudah ikut hari ini. Sampai ketemu lagi nanti. Wassalamualaikum warahmatullahi wabarakatuh. Terima kasih Bapak Ibu.
Waalaikumsalam warahmatullahi wabarakatuh.
Okay.